Okay, hemopoiesis. In England, they say hematopoiesis. Hemo, both mean blood. Poiesis means production of the formed elements. So hematopoiesis, production of formed elements of blood. This occurs in the red bone marrow. Now, the stem cells are called hemocytoblasts, blood cell formers. Blast means to form. So blood cell formers, they're the stem cells. Now what's interesting is that the hemocytoblasts, these stem cells, can become many different types of cells. They can become red blood cells, erythrocytes. They can become any one of the 5 different major categories of white blood cells. So they can become granulocytes or agranulocytes. They can become the cells that will give rise to platelets, which are called megakaryocytes. There's 2 fundamental lines, the myeloid line and the lymphoid line. The lymphoid line forms the lymphocytes, and the myeloid line forms all the other lymphocytes, or leukocytes, except for the lymphocytes, and it forms the megakaryocytes, which will become platelets. So this becomes important when we are looking at the different types of leukemia. Somebody with myeloid leukemia will have problems with making red blood cells, white blood cells, and megakaryocytes. People with lymphoid leukemia have trouble making lymphocytes, which is an immune response thing. So a very close friend of mine just recently died from myeloid leukemia. He had problems with anemia, with enough red blood cells. He bruised quite a bit because he had lack of megakaryocytes, and he didn't protect himself very well from infections, and he ended up dying of basically the flu. So what causes these stem cells to differentiate into these different types of cells? Really, there is a bunch of different factors, different hormones, etc. They tend to be called colony-stimulating factors, and they stimulate hematopoiesis. They stimulate the colonies formation. So we'll start out with red blood cells. And red blood cell formation is called erythropoiesis, making of red blood cells. To do that, we're going to need the stem cells that are differentiated into myeloid stem cells. So they respond to this colony-stimulating factors. We're going to require iron because they make a lot of hemoglobin, which has iron. It takes B vitamins, especially folic acid, and various amino acids because the other part of hemoglobin is protein, so it's going to require a lot of amino acids. If you're missing any of these, you will not get correct red blood cell formation. So the progenitor cells will undergo a lot of mitosis and make copies of themselves. They start to differentiate and they become something called a pro-erythroblast. So it means before red blood cell former. What they really are doing here is the nucleus starts to really enlarge. And what they're doing is they are making the messenger RNA that is going to be required to make the hemoglobin and the various other proteins that are happening. So the first change is starting this transcription process, and that all happens in the nucleus. They then develop into something called a erythroblast. So to become a erythroblast, what's happening is they will grow a lot of endoplasmic reticulum, a lot of ribosomes, and they will start translating the hemoglobin, and they will start making hemoglobin. They become smaller cells, but they still have a nucleus. They're still making messenger RNA, and they're starting to make some hemoglobin. As the hemoglobin amount starts to build up, we change the name to become the normoblast. Now, the difference between an erythroblast and a normoblast is that we get rid of the nucleus. Literally, the nucleus, after we have made the messenger RNA, becomes redundant. It's just taking up space that can be used for hemoglobin. So the body gets rid of it. So the difference is that we've ejected the nucleus. It becomes anucleate. And we're making more and more hemoglobin. Right after the normoblast, it becomes something called a reticulocyte, because the only thing that's left is rough endoplasmic reticulum, ribosomes, and it becomes making the hemoglobin. It's not filled with hemoglobin yet, and it's still actively making that. This is the point where the cells are released into the bloodstream and they start carrying oxygen, and they are so they leave the bone marrow into the bloodstream at this point. We know how active we are at making red blood cells by counting the number of reticulocytes. If there's an abnormally large number of them, that means that you are actively making red blood cells. If there's not enough, then, like if there's lower than expected, then you're not actively making it. Erythrocytes really become bags of hemoglobin. The ribosomes degenerate, the endoplasmic reticulum goes away, and all we are left with is a phospholipid cellular membrane filled with hemoglobin and a few other proteins to give the cell shape. Erythropoiesis, if I go back, it doesn't say it here, but erythropoiesis is controlled by a hormone made by the kidney called erythropoietin. The suffix tin means hormone. So erythropoietin is a hormone that causes erythropoiesis, red blood cell production. The kidney is stimulated to produce erythropoietin by not having enough oxygen. If there's not enough oxygen for whatever reason, then the kidney will make more erythropoietin. More erythropoietin causes more myeloid stem cells to become proerythroblasts, to become erythroblasts, to become normoblasts, to become reticulocytes, and then erythrocytes. This takes a couple of days. The reason that when people go to climb Mount Everest, they don't just go all 21,000 feet in one go, is they go up to a base camp and then they sit there for a couple of days. What's happening there is the lack of air pressure means less partial pressure of oxygen, which means that you need more red blood cells to carry the oxygen. So it takes a couple of days to make more red blood cells, and then you move on, et cetera, et cetera. And that's kind of the point. People that live at high altitude have higher erythrocyte count than people that live at sea level. People that have COPD or are breathing problems tend to have a higher red blood cell count because of erythropoietin, etc. Anything that can cause a lack of oxygen causes the production of erythropoietin. When you start to work out and you're using oxygen faster than you can deliver it, you cause more red blood cells to be made, so you can carry more oxygen, and that's where your stamina comes up. And that's part of getting in shape. It's erythropoiesis. Leukopoiesis, on the other hand, is about making white blood cells. Now, we'll see in the next video that there are different types of white blood cells. There are granulocytes and agranulocytes. The granulocytes are the neutrophils, basophils, and eosinophils. They have this colony-stimulating factor that causes the myeloid stem cells to form the progenitor cells, which then go through various things. They make myeloblasts, which then become the granulocytes, and then the granulocytes differentiate. The monocytes are first monoblasts and then become mature monocytes, etc. Lymphocytes are made from the lymphoid stem cells, and so they become in B lymphoblasts and T lymphoblasts, which will then become B and T lymphocytes, subject of a further video when we talk about immunity. They are very involved with protecting us against disease. Thrombopoiesis is about making thrombocytes, platelets. So what happens here is that the stem cells become megakaryoblast, and megakaryoblast are formed because of thrombopoietin, a hormone that makes thrombopoiesis happen. The megakaryocyte becomes this huge cell. Mega means huge, karyo means body, cyte, so huge body cell. The megakaryocyte produces all kinds of contractile elements and different proteins that are used in platelets. They make these extensions filled with this stuff that are called protoplatelets. Now, this cell gets so big that it starts to push on the walls of the capillaries of the bone marrow. In the bone marrow, the capillaries that pass through the bone marrow, and this doesn't happen in the blood, this happens in the interstitium of the bone marrow, the cells get bigger and bigger and bigger, and they're pushing against the wall of the capillaries. These capillaries in the bone marrow have got holes in them. They're called sinuses. The cells get so big that they start pushing and bulging through these holes. And the blood rushing past actually breaks off chunks of these megakaryocytes, and those chunks become the platelets. It's the same way as when you use a pasta maker to make macaroni. You push the dough out through the plate and you cut off the formed pasta. Anybody who as a kid played with Play-Doh knows that. You push the Play-Doh through the thing and you can make these various shapes of Play-Doh. It's the analogy is apt. It happens that way. So the platelets are little chunks of the megakaryocytes. So if we look at this as an overview, the hemocytoblast here differentiates into the myeloid or the lymphoid stem cells, depending on the stimulating factors, whatever is needed. The lymphoid stem cells differentiate and they become either natural killer cells, T lymphocytes, or B lymphocytes through these lymphoblasts. The myeloid branch, depending on the colony factor, either becomes a erythrocyte, so it's erythropoiesis, where you get the progenitor cell, the proerythroblast, the erythroblast, the normal blast, see the nucleus is being ejected, the reticulocyte, and then the erythrocyte. Thrombopoiesis, where the progenitor cell becomes a megakaryoblast, which becomes a proto-megakaryocyte, which becomes this huge big megakaryocyte with the little bits getting sheared off, or, depending on the stimulating factor, they become a progenitor cell, become the myeloblasts, which then leads to the granulocytes, or becomes a monoblast, which leads to the monocytes. Thus, all the formed elements come from the same hemocytoblast stem cell. The only thing that differentiates is the factors that cause changes. So you can see how the megakaryocyte pushing through the holes in the capillary gets sheared off. We will continue the next video on red blood cells.